All right, we're moving along our program, and I'm happy to introduce our next speaker, Charlotta Früchtenicht. I can't say the last name, I'm sorry. Who's coming to present from Roche, and she'll be talking about visR, a package for effective visualization in pharma. So Charlotta, please feel free to share your screen. Yep, we can see. And go.
Awesome. Yeah, thanks. This is actually a direct follow-up to Balder's great presentation. And I'm presenting on behalf of the whole visR collaboration team, which is a team consisting of members from Novartis and Roche. And actually, after we presented the idea for the package earlier this year at an RStudio webinar, we've also been joined by colleagues from other pharmaceutical companies with the idea to actually take those graphical principles and make them readily available for data scientists, especially in late-stage development, but also probably earlier in the development process, to use in their reporting. And so I'm just going to talk briefly about the motivation and then about the package, the design considerations, and the architecture. And then lastly, I will show you an example where we use visR in a time-to-event analysis. So as Balder said, there are these three principles: clarity, purpose, message, and clarity. And it is very important to use effective visualization to allow consumers of your analyses to make informed decisions. However, it actually can be quite time-consuming to create those visualizations. So looking at the right-hand side, you see all the ggplot code that goes into this one figure on the side. That is actually one of the figures in the graphical principles cheat sheet. So implementing this, especially in a reproducible way, is tedious, and therefore it's often left out in exploratory analysis. So I'm not talking about regulatory encounters. I'm talking more about exploratory analysis, both in trial analysis, but also in real observational data analysis. And often this is left out in the beginning, but then later on, if someone wants to share your figure or your table in presentations, you actually need to rework them, which is creating additional work. So actually, this was also what motivated us to team up and think of finding ways where we can make this process a bit easier. And I guess everyone who has worked with ggplot knows, and even Klaus Wilke admitted that it can get quite confusing with the themes and the scales. And so we were wondering if there is a way of taking ggplot, but also making it more amenable to implementing nice analyses in the pharmaceutical analysis pipeline. So one topic that is very important, I guess, across companies is reproducible reporting. So we know that in addition to the figures to be effective in its visual communication, there are several requirements that we have also on metadata that needs to be added. First of all, I guess visualizations should have titles and an annotation on the data source, abbreviations, statistical tests. So you can see on the right a table shell, for example, for baseline demographics table. And having to re-implement all these features every time you want to create a table one seemed like an overhead of work that we could probably simplify. So we spoke internally, both at Novartis and Roche, with stakeholders. So there were data scientists in the analytics teams, but also consumers of our outputs that are then using those for decision-making or further communicating them to their stakeholders in study design or study planning phases. And there were several aspects that seemed pretty important. And one of them is that it needed to be, whatever we were creating needed to be seamlessly integratable into our existing analysis workflows that are already building in large parts on the Tidyverse, as well as our Shiny and Markdown. And then also it needs to be flexible so that expert users can create the visualizations they want, but simple enough so that if you're just getting started, you can still create effective visualizations. As I guess all of you know, depending on who you're working with, also there might be different preferences in how outputs should be created. Should they be Excel tables, or should it be downloadable as Excel tables? Should it be a cable or a GT table? And then for some of the questions, you might want to export different visualizations to see which one's bringing across the message best. So these were the considerations, and we felt like given the importance of our in our pipeline, developing in our package is probably the best way forward. And as I said, one important part was really about the flexibility of the package, so that it can integrate in the tidyverse in our workflows. So what we came up with is this modular architecture that you can see on the right, where we wanted to separate out the functions that are used to wrangle the data from those that are used to visualize the data, as well as then those that are used to style your plot. And the latter could be, for example, then also actually giving you the ability to easily integrate corporate design colors. Whereas the first, about the data wrangling and the estimation of models, should be traceable, such that even later on, if you get in contact with your visualization results, it's easy for you to reproduce where the data came from and what has been done to the raw data to create the visualization. So we thought it might be good to follow a standard vocabulary, and since we're in pharma, late stage, CDISC Atom seemed like a good starting point to use as an analysis data frame that we can then work off of. And the idea is then actually, if we have this modular pathway, we could create wrapper functions as well, so that you can, for example, in a time to event analysis, if you want to give an overview over the cohort, you can create a plot of the attrition, how you reduce the cohort with inclusion criteria, but it could also give you, for example, a summary table of those attrition steps, or it could be a bar chart that shows you how the cohort decreased. So using the modules individually should be possible for anyone who's a more advanced user, as well as then using wrapper functions if you're just getting into it. And so just to make a bit more this more concrete, I'm just going to show you quickly a typical time to event analysis, where we start out with building our analysis cohort, and often this is shown using attrition charts. Then we want to see who is in the cohort by visualizing our baseline characteristics of the cohort, and then estimating survival or time to event. And then the first step is building the analysis cohort. I already spoke about attrition. So what you often do is you basically define who should be in the study and who shouldn't. So you might want to define a few criteria and filter your data set accordingly. And you can show that as a table, but more commonly, actually, a flowchart is used. And I don't know how many times I have actually re-implemented in Diagrammer a visualization like the one you can see on the right. So we felt like it would be good if there was just a one function way of creating that is actually also easy to comply with reporting guidelines like CONSORT and STROBE. So this one here you can actually create easily by creating the attrition table and then feeding that into in the function here on the right. Then looking at baseline characteristics, there are already amazing functions and packages that create table ones or summary tables, but usually they are very specific. We wanted something flexible that allows for different outputs, as well as gives you the way of creating your own summary statistics. So some actually like having n and percentages in a cell, and some actually want them separately, for example. And then the survival analysis, there has been amazing work done on what good graphical principles are here. And that's actually a good case where you can see this modular architecture. So we have our estimator function here, gives you full traceability of the input data set using the controlled vocabulary, so we don't have that many arguments. Then you can plot, and you can actually build up the plot. So you have the basic plot, the Kaplan-Meier plot here, but then if you want to add, for example, confidence intervals or the risk table, you can build that up step by step. And then if you wanted to change colors, add metadata, you could do that in the last step. And I heard the bing, so I guess I'm at the end of my time. We're still in the experimental phase of that package, so we're still looking for contributors or those who reached out after the webinar to reach out again. There are still design choices to be made. We're looking for people who actually have experience testing such a package, writing tests, ideas for features, or if you just want to go and look at the repository and work on issues that we have in there. And with that, I would like to thank my fantastic team, because actually most of the code wasn't written by me, but actually other people in the team. It's cross-industry by now, and I want to thank you all for your attention. You can find the slides in our repository. Oops. And we're standing on the shoulder of giants, thanks also to all the other amazing packages that have been developed before and that we're making heavy use of in visR. So thank you all, and I guess we will answer questions in the chat.
Thank you, Charlotta. I could not see any questions in a chat. So if anyone will have a question, please go on our Slack channel to ask that question. But thank you, Charlotta, for giving us a wonderful presentation about your package. Thank you.
Thank you. Bye.
Bye.